Hello, everyone. My name is Rao Yu. Rao is my family name. I work as a faculty member in School of Pharmaceutical Sciences in Tsinghua University. My expertise is medicinal chemistry and chemical biology. Today, I'm very glad to give a presentation for all of you about the small molecule-based drug discovery for COVID-19. My talk title is Small Molecule Drug Discovery for COVID-19. First, let's learn two terms about what are SARS-CoV-2 and COVID-19. SARS-CoV-2 is a virus name. It is named by ICTV. SARS means Severe Acute Respiratory Syndrome. CoV means coronavirus. COVID-19 is the name for disease. It's named by WHO. CO stands for corona, VI for virus, D for disease. 19 means the outbreak year of 2019. To make it easier for audience to follow it, I will just call this SARS-CoV-2 as the novel coronavirus in my talk. This new coronavirus is a very new type of coronavirus found in 2019. It's very similar to SARS, but it's more infectious than SARS. The best way to protect us is to wear a mask. The novel coronavirus can transmit via respiratory droplets. The typical symptoms include fever, cough, difficulty breathing, muscle pain, and tiredness. Since the outbreak of the novel coronavirus in the late of 2019, now it's almost everywhere. This slide shows the electron microscope picture of a few famous viruses. On the upper right is the Spanish flu virus. On the upper left is the herpes simplex virus 1. On the lower left is the Ebola virus. And on the lower right part is an electron microscope picture of the novel coronavirus. Here we can see some more details about the structure of coronavirus. The surface of the virus particle is encoded with those yellow color protein. It's called spike glycoprotein. And then here is the envelope of the virus. Inside the envelope is the RNA and M proteins. On this slide, we will see a general scheme of the viral life cycle. Here we can see in the case of influenza virus, the first step is the attachment. The virus will use a typically spike protein to attach the cell surface of the membrane. After penetrating into the cell membrane, the virus will start step called uncoating. Then, uncoating step will release genetic material such as RNA into the cell. After this step, the virus will start biosynthesis inside the cell, using the synthetic machine of the cell host to synthesize RNA, protein, those building blocks. Preparation of those protein and RNA, the virus will start the assembling of those together into new virus particle. The last step will be release those newly formed virus particle outside the cell and then finish a whole viral life cycle. Then we will ask the question, what kind of weapons we can have to fight against the novel coronavirus? Here we can see 4 kinds of weapons we have. First of all, the most powerful and efficient way is using vaccine. The vaccine can protect ordinary people like us to avoid the infection. Now there's quite a few the vaccine test has been ongoing in China, United States, and other countries. However, so far we do not have the vaccine yet. The second choice is using antibody. The antibody is a very powerful neutralizing agent. Usually we can find antibody in the cured patient body. The third choice will be traditional Chinese medicine, which has been proved highly successful in China. And then the fourth choice is the small molecule drugs. Actually, small molecule based drugs are the mainly available weapon for doctor to fight against the coronavirus. Since the outbreak of the novel coronavirus, a lot of efforts has been made toward drug discovery fighting against novel coronavirus. Those efforts are from pharmaceutical industry, academia research institute, and governments. Because there are so many papers has been published in peer-reviewed journals and preprint websites, I cannot cover all of them. I will only focus on 4 representative parts, which has been shown on this slide with some selected examples.